In Chapter 18, we're going to be looking at how organisms are grouped and organized mainly. So we've mentioned this person before, Carlos Linnaeus. He developed the scientific naming system that's still used today. So taxonomy is the science of naming and classifying organisms. So you have the white oak. Problems that had arose as people would call different organisms by different names, and you weren't sure if you were talking about the same thing, so they wanted to come up with a system to be able to make sure they were all referring to the same thing. So a taxon is a group of organisms in a classification system. Binomial nomenclature is a two-part scientific naming system. It uses Latin words. Scientific names are always written in italics. The two parts are the genus and the species. So, and the other thing is the first word is always written in the first letter is capitalized. Everything else in the rest of the whole name is all lowercase. If you don't write it in italics, then you need to underline the word. And again, the species is going to be a descriptor, so describing the organism. So genus include one or more physically similar species. So species in the same genus are thought to be closely related. Genus name is always capitalized. A species descriptor is the second part of the scientific name, and it's always lowercase. Always follows the genus name, never written alone. So the owl right there is Tito alba. Scientific names help scientists to communicate with one another. Species have very similar common names. Some species have many common names. So roly-poly, pill bug, sow bug, potato bug, those are all the same organism. Dandelion, Irish daisy, lion's tooth, again, same thing. House sparrow, English sparrow, mountain lion, cougar, puma, all the same animal. So those are where you end up running into issues. Linnaeus classification system has 7 levels. Each level is included in the level above it. Levels get increasingly specific from kingdom down to species. It goes kingdom, phylum, class, order, family, genus, species. The Linnaeus system has limitations. So Linnaeus taxonomy doesn't account for molecular evidence. So it doesn't look at the genetics involved in it. So the technology didn't exist during Linnaeus' time. So Linnaeus system is based on physical similarities. Physical similarities are not always the result of close relationships. So you could have, again, like analogous structures from distantly related individuals. So genetic similarities are more accurately show the evolutionary relationships. So that takes us into the modern day classification based on evolutionary relationships, and that's what is referred to as cladistics, and it's classification based on common ancestry. So phylogeny is the evolutionary history of a group of species, evidence from living species fossil record and molecular data, and it shows a branching tree diagram like what you see that just showed up there. Cladistics is a common method to make evolutionary trees, a classification based on common ancestry. Species placed in order that they descended from a common ancestor. A cladogram is an evolutionary tree made using cladistics. A clade is a group of species that share a common ancestor. So what you see right there is a cladogram. Each species in a clade shares some traits with the ancestor. Each species in a clade has traits that have changed. Derived character are traits that share different degrees by clade members. So basis of arranging species in a cladogram, which you guys will be doing in class, you're going to end up with more closely related species sharing more derived characters. And it's going to be represented on the cladogram by hash marks that you see over there on the side. So all of the organisms after that hash mark with 4 limbs, everything has 4 limbs after that point in time, and so on as you keep looking along those lines. Nodes, they represent a recent common ancestor of the clade, and clades can be identified by snipping a branch under a node. So what you end up seeing there are the nodes that are showing up. So, and it's because of those characteristics that make them different from the ones ahead of it. So right there where it branches, that is the node. And that's the derived character that they're just showing. And that's a clade. So molecular evidence reveals species relatedness as well. So molecular data may confirm classification based on physical similarities. Molecular data may lead scientists to propose a new classification. So you can see before this was how they were classified, afterwards this is how they are classified. DNA is usually given the last word by scientists. So if their DNA is close to each other, then they end up saying, well, it doesn't matter if they don't look like each other, then they have a close ancestor. Another thing is molecular clocks. Molecular clocks use mutations to estimate evolutionary time. Mutations add up at a constant rate in related species. The rate is the ticking of the molecular clock. As more time passes, there will be more mutations. So they use it to determine how closely related individuals were by how many mutations have happened since they had a common ancestor. So scientists estimate mutation links by linking molecular data with real time, an event known to separate species, the first appearance of a species in a fossil record, and so on. Mitochondrial DNA and ribosomal RNA provide 2 types of molecular clocks. Different molecules have different rates of mutations. Higher rate, the better for studying closely related species. The lower the rate, the better for studying distantly related species. Mitochondrial DNA is used to study closely related species. The mutation rate is 10 times faster than nuclear DNA. And it's passed down unshuffled from mother to offspring. And that's what you're seeing in the image below there. Ribosomal RNA is used to study distantly related species. In many conservative regions, you have lower mutation rates than most DNA. So section 4, we are looking at the different groupings of organisms, so domains and kingdoms. Classification is always a work in progress. The tree of life shows our most current understanding. New discoveries can lead to changes in classification. In 1866, only 2 kingdoms were there, animal and plant, or Animalia and Plantae. In 1866, all single-celled organisms moved to the kingdom Protista. In 1838, prokaryotes moved to the kingdom Monera. In 1859, fungi moved to its own kingdom. In 1977, the kingdom Monera split back into the kingdoms Bacteria and Archaea. The 3 Domains in the tree of life are bacteria, archaea, and eukarya. Domains are above the kingdom level, and they were proposed by Carl Woese based on rRNA studies of prokaryotes. So the domain model more clearly shows prokaryotic diversity. Domain bacteria includes prokaryotes in the kingdom bacteria, one of the largest groups on Earth, classified by shape, need for oxygen, and diseases caused. Domain archaea includes prokaryotes in the kingdom archaea. Cell walls, chemically different from bacteria. Their differences are discovered by studying RNA, known for living in extreme environments. The domain eukarya includes all eukaryotes. This includes kingdom Protista, kingdom Plantae, kingdom Fungi, kingdom Animalia. Bacteria and archaea can be difficult to classify, transfer genes among themselves outside of reproduction. It blurs the line between species. More research is needed to understand prokaryotes. So that is the end of the notes of chapter 18.